Today we are going to talk about stomata. Betel leaves are used by us for observing stomata. Different plant species have different stomata types, also the locations of stomata are different. Usually dicot plants leaves lot of stomata in the lower epidermis. Upper epidermis stomata concentration is very low. Therefore, we should get the low epidermis. When prepared, the slide water drop must be added whose epidermal tissue can be dried up. Then specimen is covered using cover slip. In this case, we have to make sure prevent air bubbles in the specimen. Stomata pores can be observed under a low power of the microscope. Now you can see low and mid-power stomata pores microscopies. Stomata are small pores found on the surfaces of leaves, stems, and other plant organs, and they play a crucial role in the regulation of gas exchange, including the intake of carbon dioxide and the release of oxygen and water vapor during photosynthesis. Betel leaves, derived from the betel vine piper betel, also have stomata. Stomata are typically more concentrated on the lower surface of the dicot leaves, which helps to minimize water loss through transpiration. The distribution and density of stomata can vary among different plant species, and they may also be influenced by environmental factors. Let's see deep details of dicot stomata and their opening and closing mechanisms. You can see rose flower-like structures in this. Also bean seed-shaped two cells in the middle of this structure, which are guard cells. Stomal spores are in the center of these two cells, as well as epidermis cells can be seen. Especially guard cells and other epidermis cells contribute to stomata opening and closing scenario. Dicot guard cells are bean seed-shaped and monocot dumbbell-shaped. The inner side of the guard cell, facing the stomatal pore, has thicker walls, while the outer side has thinner walls, as well as radial cellulose microfibers are in the guard cells. Guard cells have chloroplasts, which are only chloroplast-present epidermal cell type, and they have large central vacuole. Above things very important to stomata opening and closing. Stomatal opening and closing depends on change in turgor of the guard cells. If water flows into the cells by osmosis, their turgor increases and they expand, but they do not explain in uniformly in all directions. The relatively inelastic inner walls make them bend and draw away from each other. The result is that the pore opens. If the guard cells lose water, the reverse happens. Their turgor decreases and their inner walls become strayer, thus closing the pore. The K plus influx hypothesis explains the mechanism. During the daytime, the guard cells actively accumulate Ke from neighboring epidermal cells, thus lowering their water potential that leads to the inflow of water by osmosis from the surrounding epidermal cells. As a result, the turgor pressure in guard cells increase, opening stomata. The accumulation of Ka in the guard cells requires the energy which is provided by the transfer of electron during photosynthesis of the chloroplast in guard cells. Stomatal clossing occur by loss of K from guard cells to neighboring epidermal cells. It leads exomosis of water from guard cells. As a result, the turgor pressure in guard cells decrease, closing stomata. I think this video gave you some knowledge about stomata. See you in another video like this.